Okay. There is the URL or and the QR code. Okay. Back in business. All right, so I'm going to talk primarily about deciphering the causes of rare disease today. Really, over 60% of cases of rare disease remain unsolved when we sequence them by every approach we can come up with today. And for that reason, Daniel MacArthur and I started our Broad Center for Mendelian Genomics and the Rare Genomes Project that I'll talk a fair bit about today. Since we'll be giving a workshop on ClinGen this afternoon, I'm actually not going to talk much at all about ClinGen now, although I will give the introduction to that workshop later today. Advance. That doesn't work. Okay. So for a little context, as we think about solving the causes of rare disease, thought I'd put up a figure from a paper published a little while ago looking at the different genes and their role in thinking about their contribution to Mendelian phenotypes. And so what you see here is that for most of the genes, we have not implicated them in a Mendelian phenotype, and they may never be implicated. We do anticipate that perhaps as many as a third of genes are probably associated with embryonic lethality, and we may never have an opportunity to capture their role in Mendelian phenotype, at least in a human model. And then there's a subset where we know the gene's in there, but we can't find it. And then a small fraction, now as much as a quarter, I just put the updated stats here from the OMIM website, where these genes have been implicated in a Mendelian phenotype. So we're really working on deciphering which of these genes in the genome are in fact implicated in Mendelian phenotypes. And as I just mentioned, many of us use the OMIM website, and you can go to the statistics page on the OMIM website and get your stats at any point in time in terms of the number of genes and phenotypes that have been implicated. So to date, or as of when I downloaded this in December, 4,727 genes have been implicated in at least one Mendelian phenotype. I should point out that many genes are causal for multiple distinct phenotypes, and you can actually look at the stats here, 3,300 associated with one, but over 1,000 are associated with two or more phenotypes, as shown here. This mainly covers single gene disorders and traits, but some of these genes are implicated in susceptibility to common disease or infection, or non-disease traits, or even somatic cell genetic disease. Now, one large question a lot of us think about is, well, how many more genes are going to be identified for Mendelian phenotypes? Where are we headed from here? And actually, a nice paper from Mike Bamshad, Debbie Nickerson, and Jessica Chong looked at this question. Trying to estimate how many more Mendelian genes are yet to be discovered. And they looked at two metrics to assess that. One was gene constraints. So if you look at our NOMAD database, we identify which genes are constrained for variation, and that suggests that loss of those genes are likely to cause a severe phenotype. So you can look at how many of those genes are constrained yet not yet implicated in a Mendelian phenotype as one metric. The other metric they looked at was mouse data. How many genes have been implicated in disease in a mouse model, yet not yet implicated in a human phenotype? And if you combine those things, it suggests that there's probably closer to 10,000 more genes yet to be identified and causal for Mendelian phenotype. So we still have a fair bit of work to do. Now, one little side project that we worked on over the last few years is really generating reliable resources for the community in terms of defining gene-disease relationships. And that's one area of work for ClinGen, but there are other groups, including OMIM and many of the resources that are shown here at the top, that have been creating gene-disease relationship data resources. We also discovered that there are a number of clinical laboratories that have been curating gene-disease relationships to inform panel development for diagnostics, and they had private data sets that weren't out in the public. And so we worked to get all of these resources together and the people leading them to say, okay, can we align some of the approaches to how we're identifying gene-disease relationships and supporting their evidence develop consistent terminology for those relationships, and then publicly share the gene curations from the various groups. And then we could, when we see discrepancies in a group saying this gene's implicated in this disease and another group saying no, it's not, we can resolve those discrepancies once we identify it. I like to call this a ClinVar for genes, because essentially anyone can submit a claim, then we can bring them all together and you can see where we agree or disagree. And that this resource has now been launched on our GenCC webpage. The database is at searchthegencc.org. And we've recently published a paper and a podcast on this. And the steering committee for GenCC, so I co-chair it with Marina DeStefano, who does a lot of the lion's share of the work for this, give her a lot of credit. But we have members of all of the other resources that are a part of the group leading this effort. Now, why did we have to align on terminology? Well, if you looked at the terms that were being used to implicate genes and disease across all these resources, they were all completely different. Some used color schemes, and in some cases those color schemes were the opposite orientation, difficult. So we went through 3 rounds of a Delphi survey with the broad community, both across our groups and externally, to arrive at a set of terms for defining gene disease validity, so how much evidence there is. These are actually almost identical to the ones that were developed by ClinGen, so that actually made it very easy for ClinGen to migrate its resource, although we did add one category called animal model only, where it hasn't been implicated in human disease, but has in an animal model, and we've added that to our ClinGen approach. So to date, we now have 16,000 classifications for gene-disease relationships from 12 different groups across 4,600 genes, and just added the stats of the different submitters that have been submitting to this database. We are working on getting more, and soon we'll have an API-based submission process, and actually OMIM was waiting for that to be able to share all of the OMIM entries into GenCC. We did have to create a category called supportive to accommodate both OMIM and ORPHANET that don't actually classify genes on an evidence level. And so genes in ORPHANET, which represent the 5,330 that are here now, and then the 4,700 that will come from OMIM soon, will all be in this bucket, and that could range in any of these tiers. So that's one of the challenges, is dealing with these different data sets, some of which are curated and some of which are not, but at least it'll be a single resource that brings everything together into one place. So you can go and look up your favorite gene and see what the claims have been. Again, a reminder that many genes can be implicated in multiple diseases. So if you look at GJB3 that has 3 strong assertions, 1 limited and 1 disputed, it turns out both ClinGen and Genomic England's panel app both said strong for this skin disorder, but both ClinGen and Australia panel app said disputed or limited for the hearing loss phenotype. So this is an example of 2 different claims for this gene, one of which everybody agrees, the skin disorder, and the other, everybody disagrees with that claim and refutes it or disputes it or considers it limited. So we have been going through all of these assertions and looking at concordance or discordance, and then through the GenCC monthly calls, resolving the ones that are discordant. And so far, we've been able to agree on all of the discordances and resolve them. So now I'm going to get back to some of our specific centers' work on discovering novel cause of disease. Over the years, traditional approaches to gene disease discovery have focused on linkage-based analysis, pointing to a region of the genome and then diving deep into that region to find the causal variation. In fact, nowadays, we use mostly next-gen sequencing, and we just look at everything all at once. One of the challenges, however, that we're all dealing with is that early on, there are a small handful of rare diseases that were actually quite prevalent. And I love this paper from the ORPHANET database, where they've looked at the different prevalences of rare disease. So diseases that are extremely rare, like 1 in a million or fewer individuals with that disease, compared to some of the more common rare diseases, where you see them in 1 to 5 per 10,000. And what you're seeing here is that there's only a small number that are more prevalent in terms of their frequency in the population, but those represent the vast majority of individuals walking around with rare disease. So 77 to 80% of people with rare disease have one of these more common forms of rare disease, whereas the vast majority of diseases, Mendelian phenotypes, over 3,000, are due to incredibly rare diseases. That makes it very, very challenging to identify the causes of these 3,000 compared to what we can easily do if there's lots of people with a particular disorder. So this makes it extraordinarily challenging to finish out the Mendelian notebook, essentially. Now, as we tried to tackle this area of work, we were bringing a lot of different approaches, automation and software. I'll talk about our Seeker platform in a second. Methods development for identifying novel structural variation, RNA-seq, improved ways we annotate variants, incorporating population reference data into this approach, developing larger cohorts so we can shift from family-centric to cohort-based analyses. We continue to use exomes from a cost perspective. They're still a better first step if you're doing large-scale studies, but we increasingly are applying whole genome sequencing and RNA-seq to these exome-negative cases. And then a huge effort that we have is really focusing on ways we can share the data to democratize discovery. Our center was initially funded under the NIH Centers for Mendelian Genomics, but now, just a little over a year and a half ago, we were refunded under the new Gregor Consortium, which also includes these other centers shown here, and we all work very closely together. And our approaches are still similar to our what I just laid out, although in this next phase, there's more effort on the functional side. The prior CMGs were actually not allowed to spend more than, I think, 2% of our budget on any functional follow-up. Is really meant to sort of discover the candidates and push them out to collaborators to validate. But in this next phase of rare disease discovery funded by NHGRI, there's really a focus on the toughest cases that we have not been able to solve over the last decade and bring, innovative approaches to try to solve the unsolvable. So we continue to support a large team. Anna Donald-Lurie and Mike Talkowski are the other two PIs, and we also now working with Caitlin and still collaborating with Daniel, who started the center with me. We have a very large team focused on recruitment, curation of variation, genomic analysis, project management, development of novel methods and application of those methods, software engineers building our Seeker platform, and then a lot of focus with Mike Talkowski's group on structural variation, functional modeling and variation, and now use of long-read technologies. I think one of our largest strategies was to collaborate with everyone we could, and so we really formed a lot of amazing collaborations with our colleagues who were recruiting in lots of different disease areas, and most of our samples early on came from these academic partnerships. We still collaborate with many of these groups, and to date have sequenced over 15,000 samples and led to almost 1,000 novel gene disease candidates, 322 of which have been well supported and are either published or on their way to publication. But we still have a lot of candidates that are not functionally validated and are still in that sort of limited state. And that's a lot of what we continue to focus on today because there's still a lot of unsolved cases. We've also been partnering with patient organizations, particularly through our role in the Rare as One project that CZI funds, and partnering to recruit more families that are undiagnosed. And in fact, when we launched the Rare Genomes project, it was really to create a diverse rare disease data set for discovery, and in fact, where we could control the data sharing. Early on, there was less rapid data sharing than what we would like to see. Literally, the moment we sequenced our cases, we put them out to the community, and if somebody solved them before we did, that was okay. That was meant to faster diagnosis. That wasn't always the same view of a lot of our collaborators, and so launching this allowed us full control over data sharing and allowed which was what we felt all of our patients and participants wanted. We also did this entirely online so that it would be much easier to recruit individuals from very diverse settings across the US. And our IRB does not allow us to recruit outside of the US, but we now have participants in all 50 states, DC and Puerto Rico. 16% are non-white or Hispanic ancestry, 15% live in very rural areas, and we have the entire age range of 1 month to 94 years, many of whom are have been on very long diagnostic odysseys. So we mostly have been focused on those individuals who have had genetic sequencing but have never obtained a diagnosis. This is just a map of all of the families that we've enrolled. The orange dots are families where we have identified a cause and returned a result to those individuals. So the stats on that are we've identified either a cause or a strong candidate in about 30 or 40% of these cases. So either a diagnosis with a known gene or a novel gene, or a strong candidate, known or novel gene, or a moderate candidate, or 60% are still under analysis. Now we did segregate this by age range of the different participants. What you can see is we're making diagnoses across the entire age range. It's actually incredibly gratifying to take an 80-year-old person who's been living their entire life with a rare disease, never had a diagnosis, and return that diagnosis to those individuals. So they've been diagnosed at all ages. The other fun project that we had the pleasure to participate in is one of the KG challenges, where we took, because we have a lot of ability to broadly share this data from our Rare Genomes project based on the consent of the patients, we were able to take full whole genome data sets and distribute them through the KG challenge that Stephen Brenner and others run, and we withheld the diagnoses of these families from our submissions to ClinVar so that they could be unknown and distributed through a competition to test out different approaches to data filtration and prioritization and other genomic analysis tools. And so this was one of the highest participation of the KG challenges, and there was, our team developed an approach to score and rank the participants who were trying to identify cause of disease across these samples, many of which were actually solved by our group, and then some of which were not. Interestingly, we actually identified 2 new diagnoses that came out of the challenge itself. So not only was it fun to allow these groups to compete and look for what were the best strategies and approaches to rare disease analysis, but also to, in fact, utilize their skills to actually diagnose a couple of the cases. And we're going to continue to participate in another round of the KG Challenge to support the development of novel approaches and tools. One of the things that we've been focused on of late is really trying to better interrogate non-coding regions, and I'll just give a couple of actual cases. So this is a child with central nuclear myopathy that presented with profound infantile hypotonia, weakness, ptosis, and reflux. There was a clinical suspicion for tubular myopathy, but they were negative for MTMM1 and exome sequencing. So we had done a trio genome sequencing and added RNA-seq on using patient muscle tissue, since they had a muscle disease. We looked at SNPs and indels and structural variants and mitochondrial analyses, tandem repeat variation, and then we did RNA-seq expression outlier analysis and altered splicing analysis, and that ultimately identified the causal variant because we were able to observe the very low expression of this candidate MTM1 gene, which was the suspected underlying etiology from the start, but no variation was identified. But using both outlier expression analysis and looking at alternative splicing, we could see this skipping that was going on in this particular gene. And it turns out through looking deeply into the non-coding region within the introns, we're able to see a retrotransposon line 1 element inserted in intron 3. It's actually mosaic in the mother, and that indicates recurrence risk as well. So that was one case that was solved by whole genome sequencing and RNA-seq. Another really challenging case is a 6-year-old female with a neurodevelopmental disorder who had a variety of clinical features you can see here on the left side. And she had had an extensive workup, normal brain MRI, EEG, muscle biopsy, cytogenomic array, had normal clinical exome sequencing at GeneDx, reanalyzed every year and continued to be non-diagnostic. She'd had a normal research trio. We had analyzed her case 3 times in 2018, 19, and 2022, looked at mitochondrial DNA, STRs, and just could not solve this case. But when we applied RNA-seq, we saw that this particular case had 37 expression outliers, both underexpressed and overexpressed. And if you look at the aberrant genes per sample, it was one of the highest sort of ranked samples in terms of the number of aberrant genes we were seeing on RNA-seq expression analysis. But we couldn't find any rare variants in any of these genes that would correlate with any of the expression analysis that we were seeing. However, what we then discovered is that the overexpression was actually not due to an overall increase in the transcript, but in fact due to retained intron material that was then making it look like the gene was overexpressed. But it wasn't all of the introns, just a small number of introns across these genes that were then leading to this signal of overexpression. Interestingly, for the underexpressed genes, we actually think some of this intron retention was leading to probably nonsense-mediated decay to then reduce the expression of that subset of genes, although we don't have proof of that yet. Now, that's great, but we still had this data and we still didn't have a causal variant. Interestingly, serendipitously, one of our other analysts was looking at some cases, and this particular set of cases, they'd included a non-coding gene, RNU, for a TAC in the panel of genes they wanted us to look at, and we ended up identifying a causal variant in that gene. And then the analyst said, gee, we haven't been looking at these non-coding genes across our general analysis. Let me take a look at our cohort and see if we see anything there. And lo and behold, there was actually a known likely pathogenic variant in this RNA non-coding gene, and then found a novel VUS in the gene as well. Now, it turns out this known likely pathogenic gene is in ClinVar, although it's misannotated as an intronic variant. Again, because the non-coding genes are just not well annotated in any of the systems, honestly, we all use. So this particular non-coding gene is actually a spliceosomal gene, and it's been associated to a number of different syndromic phenotypes. You can see the different variants across the gene that are implicated. The known variant is shown here, but the novel variant is out in a region that hasn't yet been firmly implicated in many of the phenotypes, but certainly still potential could be pathogenic. Now, this particular gene encodes a small nuclear RNA that's part of the U12-dependent minor spliceosome complex. So it turns out that a very small number of introns are spliced using this U12 mechanism. It's about 0.5% of the introns in the human genome, so only about 850 minor introns. And that is why there's this small number of genes that were over or underexpressed and just certain introns that were affected across these genes. So a really nice story and correlating really well with our RNA-seq data. And we are right now collaborating with 2 groups of 2 different assays to look at the effects of this particular novel variant on splicing. We did then follow up with the family. So the phenotype of this particular family I showed you has some overlapping features. There's a lot of variation in the features across these different syndromes. They did not have any skeletal dysplasia that's so far to date been seen across all of these diseases. So if this is the right disease, then it's probably a more mild or slightly different form. But interestingly, once we returned the result to the family as our suspected diagnosis, they followed up to one of the more common features, which is an immunological phenotype where they don't generate antibodies. And it turns out this patient actually had low or no antibodies to the many childhood vaccines they had received, which is sort of a clincher that said, yeah, I think we got the right gene here. So a really challenging case, very, very long diagnostic odyssey, but ultimately solved through RNA analysis. So in summary for these cases, I would say that non-coding genes in genomic regions are still poorly annotated and may be missed by many traditional analyses. Yet RNA-seq has become one of several powerful tools that we and many others are using to aid in genome interpretation. In our hands, this work led by Vijay Ganesh has identified 23 out of 170 cases that were solved by adding RNA-seq to our analyses. So we are going to continue to strengthen our approaches and pipelines to examine the non-coding portions of the genome. And I will say, I'm happy to say that we've now added non-coding constraint within Nomad, and this is work led by Siwei Chen, working with Konrad Kaczewski, who's here at the meeting as well, so you can ask him lots of questions about this. But we've just added to Nomad the what we're calling genomic constraint, where we are annotating the non-coding portions and which ones are constrained. And it turns out 0.5% of the non-coding genome is just as constrained as the most constrained coding regions. And in fact, about 13.5% of the non-coding genome is as constrained as the average coding region. So there's definitely, regions across our genome that are highly constrained. And if we can start to identify the localization of variation in these constrained non-coding regions, it'll help us to filter noise, lots of noise in the non-coding genome from actual signal. This is still a fairly low resolution, because these are looking at 1 KB windows. However, as we continue to grow Nomad, we think we'll be able to refine the sort of windows of analysis for constraint. But that data is now in Nomad. And since every time I mention the word Nomad, somebody asks me, well, when is V4 coming? I just threw in a couple of slides to say, it is coming, I promise, it's gonna be coming early to 2023. We've had some major technical challenges with the data, because we have the largest data set ever assembled to do joint calling. We have almost 1 million exomes being analyzed for inclusion in V4, and we will bring together V2 and V3 into a single version 4, along with the 750,000 new exomes that we've been analyzing. That includes all the UK Biobank exomes. It'll all be on HG38, so if you haven't migrated, you better do so quickly, because we're trying to get off of 37. We will also combine the allele frequencies from V2 and V3. It'll all be on one, and we're updating a number of other aspects of gene constraint and ancestry terminology, doing more subpopulation inference for the V3 genomes, and a lot of QC developments that we've had to do to be able to process this data set. Okay, now to get this data set out to the community as quickly as possible, we've defined a minimally viable product to get the allele frequencies out and the basic functions that I just listed on that slide. But then we'll start adding, restoring other functionality like read visualization, text annotation, more subpopulation annotations, multinucleotide variants, our co-occurrence function, the manual loss of function curations, the Leakage disequilibrium scores. Then we'll be doing local ancestry inference. So you might have seen about a year ago, we launched this for Latino and admixed Amerindian populations, so that we could take these highly admixed populations and separate portions of their genome to assign them to respective ancestry, so we can include them in the allele counts for the pure ancestries. We're going to be doing that for the African American populations as well. And then we'll be adding verse IDs, collaborating with Alex Wagner's group, doing mitochondrial variant calling, because the twist capture exomes that are hugely populating this new data set will allow us better look at the mitochondrial genome and heteroplasmy rates, and then doing structural variant calling. After we launch V4 and it's the additional features, then we'll start working on V5, which will be much more focused on genome addition. We've generated now 350,000 genomes for the All of Us research program. The next release of that data will be coming out soon. but then we'll also include that in GNOMAD, along with, we hope to be able to include the 200,000 UK biobank genomes that have been generated, and we'll probably by then they'll have the up to 500,000 genomes, and then we'll include a lot of other genomes generated at the Broad or externally sequenced. We also are going to start working on an approach to federate GNOMAD, because there's data sets that we can't bring into GNOMAD because they can't leave certain countries. So we've been working with Daniel MacArthur, Nikki Mulder, Jordan Lerner-Ellis in Canada, Alireza Hakidi, around bringing other data sets that can't be physically moved into our premises. That's going to require some work, but it is a future approach that we're working on. So getting back to our rare disease discovery efforts, we have been comparing genome versus exome solve rates. What we've determined as we look carefully at the cases we solved by genome, and then looking, well, could we have solved them if we just did an updated exome? And what we've found is that the genome solve rate is in our hands about 12%, where we had something that could not be solved by exome. And others have found similar results. A lot of that is due to structural variants that are either too small to be found by exome CNV approaches, or they're more complex events. Also, the improved coverage of coding variants in genome analyses compared to some of the capture challenges in exome has led to about 30% of the added yield, and then deep intronic variants is another 20%, and then the tandem repeat expansions that we've been looking at are about at 10% of the overall 12% increase in rate. So that's what we've seen in our hands. So our roadmap for continuing to improve diagnosis is obviously more and more patients, but also focusing on some technology approaches, including a lot of work we're doing right now on long-read technologies, continuing with our RNA-seq and some of the better analytical methods for looking at the data. Also resolving this big pile of candidates we have through proteomics and functional studies, and then starting to look at the newer reference genomes for ability to look at regions that haven't been well delineated in the current reference genome. Also, two approaches really around democratization of analysis through our Seeker platform and other innovative approaches to data sharing. So I mentioned Seeker. This is a genomic analysis platform that Daniel MacArthur's team started years ago, and Daniel and I have now overseen the development of this platform for the last 7 years. And it's open source, so we've actually benefited from collaborating with other groups that have developed other modules that we then push into the core code. And it allows us to bring in lots of different methodologies. We now have our RNA-seq analysis integrated into this. It's also FSMA compliant, which was a requirement for us to be able to put it onto the Anvil cloud platform. It's supported by NHGRI for all of the genomic data that's being shared. So now anybody can go to Anvil and actually use the platform for free on Anvil. And we actually have had over 2,000 people from 100 countries performing analysis with Seeker. We're continuing to work on lots of new features, non-coding annotations, surprise, surprise, long-read data, also an automated reanalysis function so that we don't have to manually go back to every case, but we can find new diagnoses through automation. And then a lot more sort of user support and teaching aspects of how to use the platform and how to load your data. And so that's been, I think, really beneficial. We've had a lot of groups that have appreciated access to the platform, allowing more crowdsourcing of rare disease diagnosis. It also facilitates collaboration and data sharing. So we structure all the data, and it really allows us to share it in other venues for use. And we've shared that with Geno2MP that the University of Washington supports, and other sites, and the KG program, as I mentioned. However, just sticking data out there doesn't always lead to discoveries. How to use that data in productive ways is critical. And I mentioned earlier just how rare some of these cases are, and the only way we will solve some of these cases is through innovation and data sharing. So I'll just give you one classic example of cases that have only been solved through data sharing. So this was one of our Rare Genomes Project cases, 2 SIBs with microcephaly seizures, global developmental delay. They were non-ambulatory. We found 2 novel variants in the PPIL1 gene and that gene had never been linked to disease. One was maternal, and a stop codon mutation, a nonsense mutation. The other, just your run-of-the-mill missense. So, we've seen this sort of thing in lots of patients, not proving that this is implicated in a gene. And in fact, for many, many years, the rare disease community had a common problem. Many of these gene candidates, like the one I just showed you, would just sit in people's hands for years, never to be published, waiting for them to come across a second case that would strengthen the evidence. So years ago, we got together to define a solution of how we were going to get these candidates sitting on somebody's shelf and on their computer to actually be brought together to implicate genes in disease. And we defined this genomic matchmaking concept where 2 candidate genes from 2 different groups could be brought together computationally, and then the phenotypes compared to look for valid versus invalid matches. And so we gathered a community at ASHG now almost 10 years ago to define our goal, define barriers, which honestly were paranoia about getting scooped was the biggest barrier to sharing these candidates. And so we developed a system where if you put your candidate in, the only people that can match with it would be someone who had the same candidate. So it got around this paranoia, and both groups, the person who originally deposited it and the new querier, would both be notified by email simultaneously, bringing the groups together. And it really got around this sort of paranoia about getting scooped issue. And we developed a lot of technical approaches, APIs to connect all these databases, and then launched the platform in 2014. It's been incredibly successful in terms of really allowing many gene candidates to move from candidate to implicated in disease. And in fact, that candidate I just showed you, we stuck it into Matchmaker Exchange, immediately got a hit to Joe Gleason's group, who had by then assembled 7 other cases through some through his lab and through other matching. And we were able to just join that publication. Mouse model was also created, and the gene was clearly implicated in disease with all those cases. This platform, from the article that we just published in another special issue of Human Mutation, has documented over 1,000 novel genes discovered using Matchmaker Exchange since it was launched in 2013. So just an incredibly powerful tool, all based simply on data sharing and people being willing to enter their gene candidates in a platform. This is now used by over 100 countries, like just thousands of users, a lot of them registering through GeneMatcher at the Hopkins site so that they can not have to hook up a node directly to the platform. However, for many cases, we don't have any candidate, and we need to make broader queries of genomic data. So the Matchmaker Exchange only works if both groups have looked at their data and found a candidate, and then you bring them together. So we've identified 3 different types of matchmaking. One is the 2-sided. That's what we do today. Both groups have a candidate, and you just simply bring them together. What we want to do is really support the next phase of this, is just 1-sided matching. So one group has a candidate, but you simply want to query somebody else's genomic data and say, you might not have noticed this is a candidate in your oops, something went awry, in your data set. And so we want to just query the entire VCF file of a genomic case. And then, of course, in the future, we just want to throw all the genomes together and look for commonalities, but let's start with B first. So we're actually working on this. There's been some isolated launches of variant matching platforms, so you can actually go to Variant Matcher that the Hopkins team that brought you GeneMatcher has launched, and you can put in your variant, add your phenotype, and you will get back a list of all the patients with that variant and their phenotypes. A lot of times it quickly allows you to rule out a variant because you see, oh, it's in 4 people who are unaffected parents, or all of the phenotypes are totally different, and so this variant is probably not causal. We're now working across a number of different groups that have similar systems willing to allow their data to be queried in this way, and trying to develop the sort of platforms and approaches to do this technically. We're working with the Global Alliance that's developed 2 different APIs, a Data Connect API and the Beacon version 2 API, as ways to possibly query. So this is still in the early stages, but My goal is to basically hook up every biobank in the world and allow it to be queried and deal with some of the sort of regulatory and permission issues through some of the standards being developed in the Global Alliance, so that we can find that case on the other side of the planet that has that variant you're looking at and either rule it in or rule it out or build evidence one way or the other. Now, this platform will be tremendously helpful for rare disease gene discovery, which is what I've been focusing on, but it will also be useful for evaluating VUSs in known genes. And of course, I spend a lot of my life as a clinical lab director dealing with VUSs in that context, and our ClinGen program certainly focuses on that. And in fact, this is just an incredible challenge. There's VUSs everywhere. We all have about 200 rare coding sequence variants in each of our genomes. There are over 665,000 VUSs in ClinVar, 74,000 conflicting variants. Variant evidence is often unpublished and inaccessible, so we have to directly be able to access and query the data that's out there if we're going to be able to address this huge problem. We just did a little recent study looking at actual clinical lab data to try to characterize how big a problem VUSs are to our patient population. We collected an enormous amount of data, 1.5 million tests worth of data from 19 labs, mainly in the US, and we were able to show that 30% of all there's a percent missing there, over 30% of all genetic tests are inconclusive due to a VUS. It's a huge problem. Interestingly, we actually were able to show that a statistical drop in VUSs in genome testing compared to panel-based testing, which is a little non-intuitive. In fact, if you look at the size of the panels, as the size goes up, the VUS rate goes up. Makes sense, more gene content, more possibility of a VUS. Why then does it go down with a genome where obviously you have enormous numbers of variants? Well, the reason is, well, A, why do we even put VUSs on reports? Well, historically, single gene testing in small panels were applied to patients with a high prior probability of a diagnosis. And so often those VUSs were actually more likely pathogenic. If you had a patient with cystic fibrosis and you sequenced the CFTR gene and you found a VUS, there's a reasonable chance that was actually pathogenic. Nowadays, you take a patient with autism and you sequence a 500 gene panel, most of those VUSs are not likely pathogenic, right? So the world has changed in our current era, but we still need to facilitate the downstream steps that may be useful in following up a VUS, doing segregation studies, functional analysis, correlating with phenotype, or if phenotype evidence accumulates elsewhere, you never get it back to the patient if you haven't put the variant on the report. However, the prior probabilities have decreased, and we are dealing with a tsunami of VUSs on patient reports, and we have to do something different. Now, interestingly, that genomic data and the drop in VUS rates is actually because of the way we define what goes on a report as a clinical lab director. You can't put all the VUSs from a genome on a patient report when you do a genome. So what do? You require the clinician to give you phenotype, and you look at the evidence for the variant's pathogenicity and the match of the gene to phenotype, and you scrutinize, and you only put VUSs on that actually have a higher likelihood of being causal. Panel-based testing, basically you run whatever the physician ordered, and every variant gets classified, and every VUS gets thrown on the report. And as panels got bigger, more and more VUSs, they're often in genes that actually have almost no match to the patient's phenotype, get thrown on reports. That is why the panel VUS rate is much higher. So the challenge is a lot of these VUSs, most of them are actually going down to benign eventually. And we've been looking at some of the data. So if you look at ClinVar, we did a 3-year analysis. 5% of VUSs were reclassified, 75% down to likely benign, only 25% upgrade. The challenge is the range of certainty around pathogenicity for a VUS goes all the way from 10% to 90%, huge range. And so some of the labs, the lab I used to direct, the Mass General Brigham lab, we've been using sub-tiers, VUS, low, medium, and high, you could call them. And we have, and I'm gonna just, in the interest of more time for questions, skip through a lot of this data. But basically, we've been able to show that the favor benign side almost never reclassified up towards pathogenic, whereas the favor pathogenic ones had a double the rate of likelihood of moving to pathogenic. So using other approaches, and in fact, when we did that study with 19 laboratories, what we showed is that the 2 labs that were using subtiers, they never put a VUS low on a report for a genome, only used the higher evidence level, whereas the panel-based tests get all of these VUSs thrown on them. So now, the good news is that in the newest guideline that we are working on right now for the sequence variant classification standards, we will actually subdivide the VUS range. The draft terminology we've come up with so far is VUS low, mid, and high. That will enable more refined approaches to which VUSs we put on reports and focus more on the ones that are more likely to get reclassified. So in summary, there are benefits and drawbacks to reporting these VUSs. Strategies are needed to reduce the unnecessary follow-up and patient anxiety, yet still facilitate variant reclassification and delivery of new information to patients. So we're really working on new guidelines. I actually am about to form a new working group of ACMG to focus on this guidance. This is what we've put into the paper that we're trying to get published right now, is really encouraging more clinical data, not just for genomes, but for panels, inclusion of parental samples so that you can find de novo and phasing information directly, even for panels, utilizing the VUS subtiers, creating supplemental sections of reports, so those VUSs are not front and center, but they're a little more buried if they're less clinically significant, and allowing physicians and patients actually to opt out of VUS reporting, particularly in cases where the genetic suspicion is actually quite low, like in a lot of cancer testing, for example. And with that, I'm going to stop, and I'm happy to take questions. This is many of the people in the team that have worked on a lot of the projects I mentioned here, powered by our newly found Nomad beer. The team loved this beer brand, and this is a link to the slides if you want any of the slides that I've shown. With that, I'm happy to take any questions. Thank you.
Yeah, I was interested in your comparison between the yield of whole genomes and panels, and your explanation makes sense. But I was thinking, maybe, are we not comparing apples to oranges? Because sometimes we do panels for screening with asymptomatic patients, and the need to report variants is very different when you do rare genetic disease, where there's phenotype or should be phenotype. So I just find that this may continue when the panel application is. Those tests have different applications. That's the only thing I want.
Yeah, it's a good question, and I didn't clarify. For that particular study, we only collected data from diagnostic testing, and we excluded all population screening, carrier testing, all of that. So it was only looking at patients who were symptomatic and the testing for it, because that's absolutely a good point. And we had, in the version of the paper that's posted on medRxiv, we also included not only the VUS rate, which was the point of the paper, we also included the yield and showed that genomes have higher yield than the panels. The challenge with that is it's not comparing apples to oranges. These are 2 different patient populations, and the first round review validly pointed out that it's just not useful to do that comparison when you're not taking the same patients and looking at genome versus panel. So we actually, in the current version of the paper, removed the yield comparison and just focused on the VUS aspects. Question in the back.
Antonina Mitrofanova, Rutgers University. Thank you so much for a beautiful talk and absolutely fantastic initiative. Are you planning to expand it to include any epigenomics data types? And if yes, what kind of epigenomics would you do?
Yeah, it's a great question. It is something that we have talked about in terms of methylation studies and other approaches. We have not yet started that. So we don't have specific plans at the moment, but it is something we've talked about, is starting with methylation analysis, basically, to look at epigenetics. But right now, we've been more focused on the RNA-seq, long-read, and some of the other stuff I mentioned. But it is in the list of things to do, so it's a great question.
Thank you so much. It's absolutely fantastic effort.
Thank you.
Hi, Heidi. That was a great talk. Sean Mooney, University of Washington. So I have a question about function. And we know that function kind of sits on a spectrum, that some variants completely knock out whatever the biological function is that is giving rise to the disease. And some variants, maybe partially functional, like will have some kind of a small level of function, and some people will cause disease and other people will not cause disease. And I guess my question is to you is kind of your thoughts on how do we, I think a lot of variants of unknown significance are likely to be these kind of things in the middle that are, maybe partially functional, and in some patients, maybe a few patients, it causes disease and is pathogenic, and in another patient, and maybe a lot of other patients, it's benign and has no real effect in terms of, what we see clinically in that patient. How do we kind of get at that? Like, how do we start to understand that, a certain, Mendelian inherited recessive variant is, maybe partially functional and is maybe only going to be, cause a clinical phenotype, even though it's pathogenic, maybe only cause a clinical phenotype in like 5% of the patients that you observe it in, or something like that?
Yeah, it's a great question. I mean, personally, I think most of the variants we find probably have no effect. But I do think you're absolutely correct in that there's probably a lot of variation that has some effect that we're just not able to capture, or some of the work Tuli Lapulanian has been doing in looking at allele-specific expression. So maybe in some individuals that variant's causing disease because that allele is overexpressed, or in others not causing disease because the wild-type allele is overexpressed. And so it's actually a different expression level of the two respective alleles. Could be one of many different, examples. In other cases, it could be a different locus that's somehow interacting and contributing to the differences at another variant at another gene locus, or something that's a modifier. So I think there's going to be a lot of different explanations, and I think we're going to need to actually share the full genomic data sets to be able to look at modifier loci. But doing that, like, I had 5 cases and I looked at 5 genomes, that's not enough to show these smaller effects. Also, hypomorphic alleles, just simply the alleles are going to give a different phenotype just because they are not as severe. You mentioned different levels of impact. Is it a full null, or is this a reduction in expression or enzyme activity, or various ways you might have a hypomorphic allele? And already we are sometimes challenged in these auto-classification algorithms that take variants based on allele frequency and throw them out. And then if you look at larger populations, you discover some of these alleles, when you look at a more statistical population scale analysis, actually have small effect on either individual phenotypes of a syndrome or just simply milder versions. There's many examples of that. So I think we're going to both need to look at the rest of the genome to look for modifiers, be able to look at allele balance and expression of these genes, and also do more statistical case control analyses to look at hypomorphic effects and things. And the list probably goes on of other approaches to try to tackle this great challenge, I think.
Do you see a pathway where we might be able to annotate hypomorphs better in the databases that we look at?
Yeah, it's a good question. We've been having discussions about terminology for these variants, how to annotate them. My view is I like to call them, to separate pathogenicity from penetrance, also then separating the mechanisms of penetrance, right? So you might say pathogenic with low penetrance due to a hypomorphic functional state. Other things are lower penetrance having nothing to do with being a hypomorph, they're just low penetrance. So what I would like to see is other annotations on variants. And some have said, well, just it's use a different term than pathogenic. Well, no, like pathogenic is, in my mind, can it cause disease? Not will it cause disease. That's more penetrance. And then how does it cause disease? And being able to characterize things like hypomorph or other mechanisms. Is it gain of function or loss of function? And different ways we think about a lot of different ways. And there's a lot of complexity into the mechanisms of pathogenicity that still need best terms for.
Great, thank you. Impressive talk.
Thanks.
Yeah, hi. It really was a great talk, so thank you so much. I wanted to go to a basic question about genome sequencing accuracy, because obviously you would be incredibly sensitive to changes or differences in accuracy of these genomes. And I remember, 8-10 years ago, people were publishing, you publish this, if you do the same genome twice, even on Illumina with 30x coverage, it was something like 20,000 variants either appear or disappear, something like that. I'm wondering, where are we in terms of accuracy, and where does it affect some of these analyses? And especially when we start thinking about all the different ways of sequencing, are any of them looking to you to be dangerous? You wouldn't want the families to come with you and say, oh, we already did our genomes at so-and-so, and here they are. So just any musings about accuracy?
Yeah, it's a great question, and there are still major errors in the best sequencing technologies. They obviously vary depending on whether you're talking about single nucleotide substitutions in diverse regions of the genome versus homopolymer regions. So there's lots of different factors, whether it be the actual sequence, the mappability of the data. The sequence may be fine, but then the sequence exists multiple regions, so your mapping is challenged. We're getting much better coverage, evenness when we use PCR-free genome approaches, but still, for cost reasons, we often only sequence, at 30x coverage, and there will be regions that are challenged. And also, as we think about mosaicism, and low allele fraction, like capturing that accurately is obviously going to be very challenging. As we look at copy number variation and structural variation, depending on the size of that, there are lengths that are just challenging, for the different approaches that we're taking. In order to accurately capture structural variation, a lot of groups use like 10 different, methods, and then look at which ones said the same thing across all of them. Like, we're still at early stages in the accuracy of certain types of variation and certain regions of the genome that are more challenging for one reason or another. It's been exciting to see some competition in the marketplace, but I can tell you a lot of these technologies are still challenged in having the same accuracy, which still isn't perfect for our best technologies today. And then they're further challenged by the fact that we use, resources like Nomad that are widely used. There's no reference genome data set for the new technologies, and who's going to pay to build up that data set? Same is true of long-read sequencing technology. There's a consortia being formed right now to try and pool long-read data that's being generated by multiple groups, so we have better reference data sets for comparison to rule out artifacts that are across these data sets. So I guess long story short, we still have a ways to go to be accurate across much of, aspects of our genomes. Though I will also say, more excitingly, it's amazing what we can do today and the data we can generate. So there's the basket half full or half empty as you look at it. We do still have a ways to go on accuracy, but we do have great technologies today. Yeah. Two more. Go
if
ahead.
I may. So I think the VUS discussion is interesting because, as you pointed out, the VUS report could be useful to follow up, but it's also a little bit, potentially dangerous if people, especially primary care doctors that may not fully understand all the nuances, may recommend, say, procedures like a double mastectomy by VUS, right? We all hear those stories.
Yeah.
But I was wondering if also, how, having some sort of guidelines to report VUS would be great, because I see it's very variable between labs. And I was wondering if those guidelines could also be impacted going back to the application. For example, if you're doing a screening test, or you're doing a rapid newborn genome, you may be more tolerant in the second case, because once the medical action that you're going to take could be life-saving, versus in the screening, maybe the action maybe not really that well thought of, right? So I wonder if when you're thinking about this reporting, you could also think about the different applications of genomic testing.
Yeah, I think these are all good points. A lot of the reason we put VUSs in reports today is it's like a one-shot opportunity to get information back so that somebody could then track and follow that VUS. And patients are going to ClinVar themselves and looking things up, and the physicians go look things up. I think if we had a more robust infrastructure for maintaining genetic data and the electronic health record and updating data, then there would be less of a feeling that we need to put all the VUSs out there today because it's our only shot to communicate, versus when we learn more, then we'll issue an updated report. And we'll nuance that based on the question you're asking, what the options for follow-up are. Is this like a VUS in CDH1 and I'm going to have to remove your stomach because of cancer risk? Or is this a easy, quick enzyme assay to confirm a phenotype, right? And those are just like very difficult to delineate those different scenarios. But I think education of physicians and better clinical decision support tools in our electronic health systems to help guide clinical decision making, and in fact, what kinds of follow-up to do, are all things that eventually could help guide this space better and reduce some of the risk of inappropriate decision making with VUSs based on just a lack of understanding of how little we know about most of these variants. So I think it's a great point, and we are going to try to come up with some guidance based on today's infrastructure, but that's going to have to be updated as we develop better infrastructure over time. Yes.
Yeah, and this is also a question like Russ's where I want you to sort of speculate, but one of the things that's been interesting to me over the last 20 years of this work, which you've had a cat's eye view of, is how the improvement in phenotyping, particularly standards around phenotyping, have made possible these big international consortia, your ideas for the extension to Matchmaker. So would you tell us a little bit about how those changes in phenotyping and phenotype standards have affected the work and what remains to be done?
Yeah, phenotype is a huge challenge, and we deal with it on multiple levels. One is defining disease names, and like ClinGen has a whole lumping and splitting committee that tries to delineate, are these distinct phenotypes or distinct diseases that we're classifying valid relationships on, and how do we delineate them? Are there different mechanisms by which pathogenicity leads to disease, versus just a spectrum of phenotypes that's not due to any pathological or pathogenetic mechanism. But more getting at your question, as we look to scale and find novel causes of disease, determine what these VUSs are, we definitely need to gather more phenotype data and gather that data at scale. Certainly the use of the human phenotype ontology has been revolutionary in terms of rare disease phenotyping. It is the standard that groups are using today. Interestingly, there's been some studies showing more terms in phenotyping actually do not lead to improved diagnoses. Now, it's a little counterintuitive, but an argument is, well, some of those phenotypes might be secondary, not critical, different things going on, and if you use some clinical judgment to say these are the 3 key phenotypes that I'm really looking to associate a causal etiology to, and I don't think these 7 other things going on are probably related. Now, there's some judgment there, but it may be why the added extra phenotypes are actually creating sometimes more noise than help in making a diagnosis. So I think it's a tricky space to think about the best way to do this. And also, pertinent negatives are often not effectively captured, which can be really critical in ruling in or ruling out phenotypes. Also, a lot of the phenotype prioritization algorithms that are being built into the genomic analysis tools are based on fairly old and static data sets, like the first publication that was cataloged in OMIM. It is not based on a series of patients collected and diagnosed over the last 10 years, right? We need to drive our tools off of real data from real patients, because every time, after that first discovery and then the next, 100 cases come out, those phenotypes often migrate a bit to other more severe, less severe, different things. And so getting real case data to inform what the phenotype for a given gene-disease relationship is will be critical. And I give credit to the Decipher database in the UK, where they have been populating thousands of cases in there with the diagnoses and the HPO terms. And I've been talking to Peter Robinson, can we drive Exomizer and Lyrical, his new tools, off of real Decipher data instead of cataloged information in OMIM and other resources, because I think that will make these tools more accurate in the use of phenotype to diagnose both with known genes as well as make novel discoveries. So a few thoughts there. It's a complex area, but I think it's going to be critical to really utilize phenotype better than we've been doing to date. Yes?
Amazing talk. Many people here are involved in machine learning and AI type of research. And so I was wondering, from your perspective, what do you think about the overall effort that is invested into the clinical or variant type of interpretation? And where do you think these resources and new algorithms are most needed, and how is this landscape looking?
Yeah, I think, like you can never do much in the way of machine learning or AI if you don't have the data and you haven't structured it and standardized it. And so, I really want to move our community to getting the raw data out there in ways that are structured and standardized. Phenotype, as we just talked about, is critical, but other annotations on that data and the evidence used to call things pathogenic, use of the ACMG codes, but the underlying data that led to the application of those codes, I think these are ways that we'll be able to better use machine learning if we can have that data standardized and structured. And we'll talk a little bit about that in the workshop later today for ClinGen, as we gradually move towards better structuring and standardizing of data, because that is what's going to fuel a lot of this work in the future. Thanks. And I think with that, it's time to, have lunch or something other interesting thing. Thank you.